and welcome to today's lesson, which is a continuation from what we have been studying. Now, last time we were here, we were talking about coordination in animals, and we were looking at an organ called the eye. We had seen various parts of the eye and what role do they play in helping us in achieving the function of the eye. So eye is a very specialized organ for us that is really modified to help us achieve the actual function of vision. Now, the last time we were here, we were seeing how an image can be formed in the eye. So we were talking about formation of image. Now, this image is what will be helping us to actually visualize what we are supposed to see, what we are supposed to be looking at. And then you say that I have seen a goat, I have seen a cow, I have seen a human being. So that image of the human being, the image of the cow that is before you, must actually be formed in a part of the eye that we looked at last time, and then the information must be taken into the brain so that it is interpreted, so that you say that I am seeing such and such. But how was this one to be done? Now, we say that an image is always formed in the part of the eye called the retina. So we say that image is formed in the retina. This is the part where it is formed. Remember, we were saying that the retina has got two light sensitive pigments, that is the cones and the rods, and we saw how they were distributed in the eye. So for this particular image to be formed, that particular object must be reflecting light, so that this particular light reaches the eye. And when the light is reaching the eye, this particular light must be bent in a way for it to be focused on the retina, or for the image to be focused in the retina, or to be formed in the retina, that is. So this bending of the light comes if light is traveling on different media with different refractive index. So this light must be bent or must be refracted. So the first part of the eye that refracts the light is called the cornea. That is what we saw there in front part of the eye. So it's a medium which will change the speed of light when it is passing through it as compared to when it is passing through the air. So we start from the air, the light traveling from the air and then reaching the cornea, and then it goes to the lens before it reaches the retina for image formation. So that is how the image actually will be formed. That light first of all is bent through air, and then when it is reaching the cornea will bend it further so that it can be focused. And then behind the cornea there is the lens that will further bend the light. So that is what we were saying last time, how the image is really being formed. So that is what happens first: the bending of the light through the air, then the cornea, and then the lens, and then actually the image will be formed. So this one happens because this particular media here have got different density. So you see that the object, this is what happens: object reflect light towards the eye, and then in the eye, cornea bend light, or we can just say bend light. So this aspect of bending of the light is called refraction. So you can say refract light. And then the next thing is lens will also bend it further. Will bend it further. And I've said bending means refract. Okay, so light will be refracted further. This is what is leading to image formation, leading to image formation in the retina. That is what we learned. Now, just a little bit of the reminder: this particular image is actually formed when it is upside down on the retina. And we said that, I have just said that retina has got pigment. Retina has light sensitive pigment. light sensitive pigment. These are rods and cones. Now we have just stated sometimes back that this particular light sensitive pigment, they perceive light of different intensity. So we said that the cones will always perceive light of high intensity. At the same time, they are able to differentiate the colors. The rods will be perceiving light of low intensity. So in terms of distribution, they are distributed all over the retina, with the exception of part of the retina called fovea. Fovea is part of the retina, but it has got high concentration of cones. So in this part, there is no rods in fovea. So there is only cones there. So at that part of the retina, we can only see this particular pigment, which normally perceive light of high intensity. And then once this particular image has been focused there in the retina, the information should be transferred to the brain so that the brain can interpret. And that is why sometimes we say that the brain is what is seeing. Actually, it's the one that interprets, so that we are able to visualize the image the way it is on the real life situation. Because in the brain the image will be actually upright, but before the impulses will be leaving the retina, there is a part of the retina that we call blind spot. There's a part that we are calling a blind spot here. This is a part of the retina that is just at the neck of what we are calling it is at the neck of what we are calling the optic nerves. Now the blind spot is called the blind spot because it is lacking the light sensitive pigment. So we can say blind spot, it lacks rods and cones. So that is why it's called a blind spot, because there is no image that can be formed there. So that is one key thing that is helping us to understand why we are calling it blind spot. So this is actually how image is being formed in the retina, and those are the events that take place when the image is being formed. Now I want us to understand some aspect here. That is the iris. There's this part of the eye that I want us to see what happens in it. We call it iris, and we want to see how it helps us in the focusing of this particular image. Now we have studied the parts of the eye, and we have seen what each part of the eye should be helping us with. So we want to see the iris reflex. Now this particular iris has got a role in making sure that it controls the light that is passing through the pupil, so that the image is going to be focused. So sometimes this light can be in excess, and this particular excess light can actually damage the light sensitive pigment, which is the rods and the cones. So too much light can destroy the cones, it can destroy the rods. So it has to be regulated. At the same time, little light or less light reaching the retina will make the image not to be sharply focused, so it will be blurred. Okay, that is the image that is not actually focused in a clear way. It's not going to be clear. So therefore, the amount of light that is passing through the lens, that is through the iris, must be controlled so that we get the correct amount of light that should be passing. Now, how is this one achieved? Now the iris has got some muscles that is connected with, that is going to control the eye. So let us assume that at this part here, the one I'm drawing here, let that one be the pupil, and then we have got this particular muscles here around it. We are calling them circular muscle. These are muscles that are found around the pupil of the eye. So the first one there is there, then we have got these ones, then we have got this, then we have got this. So this is how the iris is. This is what the picture we are having. This is actually the pupil, that is where the light will be passing through. And then these particular muscles here, we are calling them circular muscles. That is the name that we are giving to them. And then there is this particular muscles here that we are calling them radial. They radiate from the center here. These ones are called radial muscles. We call them radial muscles. So these two types of muscles help in the controlling of the size of the pupil that will, at the end of the day, control the amount of light that is passing to reach the retina so that the image is focused. So sometimes we have got various light intensities and we can shift positions. Like, for example, let me assume that I am in the bright light. Assuming that bright light. Let us understand this situation here. So we want to see how these particular things will behave when we are in the bright light. So actually, what do you understand with the bright light? This is now a strong light. And remember, there are two instances that we have talked about. I have mentioned that if light is strong enough, it might destroy the cones and the rods, especially the cones in the retina. So it means that excess or strong light is dangerous to the eye. So there is how the eye will behave to make sure that it is only the correct amount of light that is reaching the retina. So I am in the bright light, assuming that I am from the dark place, and then I abruptly go into the bright light. There will be changes in the eye automatically, which will tend to regulate the amount of light that is reaching the pupil. So how will the changes of the eye occur so that they can give us a path through which this particular light will be passing? So this path that the light will be passing so that an image is formed is now what we are calling the iris reflex. How it is going to behave so that we have got the correct amount of light is passing. So the first thing that will happen here is that the circular muscle are going to contract, this one here. So you can say that in bright light, the circular muscles contract. They are going to contract. So when they contract, this one will make these other muscles to behave in a specific manner. So when they are contracting, the radial muscles will relax. So you can say radial muscles relaxes. So these are the events. This is what is actually happening. Circular muscles, they are contracting and therefore they are shortening. That is the thing. And then the radial muscles will relax in this particular situations. So when this particular two behavior comes in, this is going to make the size of the pupil to become small. What we are calling it constrict. That is what's happening. So we can say the pupil. Remember, the pupil is the hole through which the light is passing. So we can say the pupil constrict. And I've said this one means become small. Become small. Then obviously when the pupil is small, you can tell what amount of light is going to pass. So automatically, small amount of light will be allowed to pass. And therefore, this one will prevent the damage that could have been caused by the bright light on the cones on the road, on the retina. So that is how the reflex acts here. The iris reflex is going to control the amount of light that is getting in, and it prevents the damaging of the cones. So you can say these allow allow moderate light to pass through the pupil, to prevent the damage, the damage of retina, or you can say the light sensitive pigment on the retina, the light sensitive pigments on retina. So that is one thing that will help us in regulating amount of light. Now equally, we can also realize that when you are in a dim light, remember when we can talk about the situation when the light is dim light. Now one thing that I want you to understand, there is always the concept here. There is that biological knowledge that should focus. You should not lose focus on. Like for example, our focus was what are the changes that occur on the eye to prevent the damaging of the retina. That is, if any case it was not controlled, the too much light will have a damaged retina. So that's the thing. So the events here will be making sure that there is a small amount of light passing so that there is no damage. Now in the dim light, what is the problem here? That is the first thing that we should be trying to understand. We should be making that one to become our theme, to become our center of focus here. That when the light is dim, actually the normal thing, the common sense thing is that you will not see properly. And why don't you see properly? You are not going to see properly because the image is not being formed so that it can sharply be focused in the retina, then it can be interpreted. So we want to see how we can try to make sure that there is that sharp or clear focusing of the image in the retina whenever light is low. That is the dim light. So the first thing that we should be struggling with is that at this point we want to encourage more light to reach the retina. That is the thing. We want more light to reach the retina. But what is preventing light from reaching the retina? The amount of light that will be allowed to pass through the pupil. So by this time we must increase the size of the pupil. That is the key thing. So the same thing here. So the pupil here must be big enough. So if I was to draw it, I have to draw that the pupil here, if this is the pupil, I have increased the size as compared to the first one there. So this is a bigger pupil. So these are still my circular muscle here. These are my circular muscle. They're like that. So you see, this is called circular muscles, and then there are also the radial muscles. We have got these other muscles. So these ones we call them radial muscles. So these are the two muscles that we have. Now these particular muscles, they are the one to control the size of the pupil. So in this time, they are going to behave in the opposite manner as they were behaving when the light was bright. So circular muscle, what will happen to it in the dim light? Circular muscles will relax. That is the first event. Then the next event is the radial muscle. Radial muscles will contract. Now those two happenings, or those two motions, will always make the pupil to dilate, that is, to become big. So the event here will lead to these two motions. These two motions makes pupil to dilate, that is, to enlarge. So you see what has happened here. The muscles dilate, the pupil dilates, so it's widened. So that one means that it can allow more light to be passing at this particular time. So more light will be passing And when there is that particular, the increased amount of light passing to the retina, the sharpness of the image that's going to be formed will be increased. So there is that particular clear image being formed in the retina, which will be relayed. The information will be picked from the retina by the optic nerves. Remember, optic nerves is just at the section of the retina. They are still the same point. So it will pick the information to the brain for interpretation. So that is how the reflex of the eye help us in actually making sure that the image is visualized. So maybe in terms of the transmission of the impulse, maybe we can draw and see what are the stages, what happens from which particular point, and then how does it move until we respond. Because we have seen that in this particular response and coordination, there must be two things that we are trying to understand. The first thing that we were understanding that is that in this particular chapter, we have got what is called the stimulus. Apart from the stimulus, there comes in coordination. And then after coordination, we have got the response coming in. So these are the two things, these are the things that are there. So in terms of the events, how these particular things unfold, maybe we can try to have a clear way forward and see where is the stimulus coming from, and where does it go to, and then what happens next, and then like that. So maybe we can start from that particular object here until it is reaching the iris. What normally happens. So the first thing is that the response is the stimulus is the light. So we have with the stimulus. So here we can talk about light. So this light must be coming from the object. That is the first thing that is happening here. And then once it comes in, it will be received. So whatever is receiving is called receptor. And in this case, the retina. It is going to be focused into the retina so that the retina will be receiving. So actually the retina here is actually acting as our key sensory organ here. So that is the first thing. Then once the receptor has received the information, we said that it will be picked up by the nerve cells, the neurons that will be transferring them. So we have got the sensory neurons that are picking it. So I have got sensory neurons which are found actually at the optic nerves. So we find them at the optic nerves. So that is the next event here. After bringing in the stimulus from the light, we have seen how light is reaching the retina. That's what we have been struggling to talk about. And then from the retina, we have got the sensory neuron that is found in the optic nerves is picking up the information. And then it is going to take it to the unconscious part of the brain. So we take it to the unconscious part of brain. So we are going with this particular information to the brain so that actually it is going to be coordinated so that we can interpret it, and then we can understand what's happening, how the image is going to be formed in the right way, upright way. So that is what happens there. From here, we are taking the information by the motor neurons. So the next things that we have got, the next event is that the motor neuron is picking the same information. So it's a nerve cell in the iris, motor neuron in the iris. We are taking that particular information back so that we can really respond to it. So the iris here has got muscles. So the iris muscles here that we have been talking about, the circular and the radial muscles, they'll be acting as our effectors. This is what will cause the action. These are our effectors. And then when these ones occur, when that information comes back there like that, the effector here means that we are going to form an action. We are responding to the situation. And the response here will come in terms of the changes that will be occurring in the size of the pupil so that we can see what amount of light do we accommodate to pass. So we can talk about, after this, there is response. And in the situation that we are talking about, the response is brought about by the size of pupil, so that we can see the amount of light that is allowed to take place. Like when you are in a dim light, the size of the pupil, how will it be? It will be widening, it will be dilating. So that is the actual response there. If you are in a bright light, the pupil will constrict so that it becomes small. That is the response, how you are responding. So these are the events, how they move from the time the light is coming in until the size of the pupil is controlled, is controlling the light that is coming in. So that is a response that normally occur in our body. So this is what we should be understanding up to this particular point. So that is the next thing that I wanted you to understand, and I think it's a very simple idea and straightforward, and if you follow it very well, I know everything will be okay with you. Now I want us to have a look at this particular aspect of accommodation here on the eye, so that we see how we can make sense out of it. Now, accommodation, actually we are trying to talk about the changes that take place in the eye which allow us to see object from different distance. Like we can talk about, you can see an object from near point, we can see object from far point. Now, during the time that we are trying to focus this particular object so that we can see well, there will be some changes that will be taking place. Like assuming that you are using the camera, either of the phone or the normal camera that we have, for you to focus on something, there is how we normally behave with the camera so that we can take a good picture. Sometimes you can change the position of the lens to produce outward so that we can capture it. Sometimes we can lower the lens so that it come in the camera so that we can actually focus. Now, that is actually how our eye normally behave for us to be able to see the object very clearly. So that aspect of changes that take place in our eye is what we are calling an accommodation. And the eye has got cells that is enabling them to behave like this. Like for example, we have got some cells that are protein in nature in the lens here that can enable them to change the shape so that we are able to focus the object. So I want us to see how these particular changes can take place so that we can understand. So I have got two objects that I want to focus on. There is a distance object, and there is also a near object that is closer to you. Like maybe you are reading a book. So how do you normally see? How do the eye normally change so that we can actually have got a clear vision? So look at this particular diagram of mine. The object is far from the eye. So the object is reflecting the light. So when the lights are reflected by the object, when they are from a distance, it's like they are coming parallel with one another. So for them to be focused in that particular manner, the lens will divide, will bring those particular lights together more if it is more convex. So since this particular light is coming when they are from a parallel direction and widespread, they are coming closer to one another, we don't need to bring them, we don't need to struggle so much to bring them together. So what we need to do at this particular point here, we have to make sure that this particular lens is less convex, meaning it should flatten up so that we can allow this particular light to not to bend so much as we are suspecting. Now, how will this one happen? Now, I want us to look at it in terms of how the pupils will be behaving in this particular way. So we have got the circular muscles that are holding the lens. So these particular circular muscles, at this point, they will relax. And then the suspensory ligaments that are holding it will be pulling, they'll be tightening. Now, when this particular eye behave like this, there is a fluid here. This place is not empty. So this particular fluid here will be having pressure being focused on the lens here. So the lens is going to have pressure, and that particular pressure will be making the lens to appear to be flat, and therefore it will be enabling it to converge this particular lens, slightly converge them so that the image is focused. So that is what we have seen. The behavior of this particular the muscles here, the circular muscles here that we find that are holding the lens here, they are going to relax. So when they are relaxing, they are very long muscles, they are going to give this particular muscle, that suspensory ligaments to at least to pull, so that they are going to be tight and they are contracting. So that is making the lens to have got pressure being building from the fluid that we have before it, to make it appear to have got that flat shape, so that it can bring in the rays together and focus them in the retina. So that is what happens when the object is from a far off distance. Now, when you are having the object from a near distance, what is actually happening? Now, assuming that you are holding the book from your hand, what normally happens is that that particular book is already reflecting light from one point, because they are close. And when it is coming from one point, it means that rays have to spread. That is diverging. So when it is diverging here, they might not even be in a position to reach the lens very well. So when they reach the lens, these are rays that are already spreading out. So what we need to do, or what the eye needs to do, is to bring them together so that we can focus a sharp image here. That is the thing. From near point, they are already diverging. Unlike when they are from far, they are coming when they are like more of parallel in direction. So when they are already, like you can see, if this is the point where the image is, the object, I mean, the image, the light that is reflecting are like spreading out. That's what we are calling diverging. So we need to really converge them completely. That one can be done when the lens is more convex. So when this particular rays reaches retina, when they are from near object, the retina must be more round, more of convex, so that this particular, it is able to converge this particular rays into the retina, so that we can focus this sharp object, sharp image. So what will be done here is that the muscles here will be helping us to change the shape of the lens, so that it can become more of a convex. And this is done when the circular muscles themselves, now at this point, they contract. So their contraction is enabling the suspensory ligaments to relax, and therefore when they relax, they are long enough in shape, in size, so they can slack, they loosen up, so that they have got that particular more room to accommodate these particular circular muscles. Now this one here will be making the lens itself to become more of round and convex. So when the lens is more of convex, it is able to diverge the rays together. It can bring them together very easily, so that it can form the image. So these are the things, these are the events that normally take place all the times that we are trying to see objects, whether from near point or far point. These are the changes that take place, so that we are able to visualize the image and then focus them very sharply. So all this is called an accommodation, the changes that take place in the eye, so that we can see object whether from near point or from far point. That is what we are calling accommodation. So that is what happens, and I hope you have understood how the changes take place and how we can focus the object clearly